Welcome to this Thrust Group 3 meeting. Today, the only speaker is from Michigan State University, Mike Weiser, who's a grad student in Rich Lenski's lab, and is going to tell us about fitness trajectories.
Yep. If we could mute the distance locations for now, that way we won't pick it up and send it off to everybody else as well.
Okay. All right.
Alright, so my name is Mike. I'm a grad student in Rich Lenski's lab, in the process of writing a dissertation at the moment, so I'm talking to you today about some of the work that is in my dissertation. The first half of it is going to be primarily about work I have done with long-term lines with bacterial wet lab samples, although I have information about digital work on the same general principles at the end of the talk. So, my interests are primarily in the predictability and the repeatability of the process of evolution itself. There are a lot of useful things about evolution, but the primary trait, or if you want to argue if it's a trait, we can get into that later. The primary factor that is being optimized within the population in the evolution itself is fitness. And for the biologists in the room, we know what we're talking about. For the computer scientists, fitness sometimes means something a little different. So we are discussing fitness here in an evolutionary sense. Now, a couple of individual examples. This guy over here is Michael Phelps. He is an incredibly talented swimmer. He has more gold medals than anyone else ever in the Olympics. He has more Olympic medals in general than anyone else. In fact, he has twice as many gold medals than anyone else has ever had. On the other hand, from an evolutionary sense, he's not particularly fit as he has absolutely no children, and we do not yet have any evidence of his inclusive fitness managing to rise by providing resources for close kin to reproduce. On the other hand, we have this family over here from Arkansas who have 19 biological children. From an evolutionary sense, these people would seem to have a much higher fitness. But of course, measuring fitness in any sort of realistic population is difficult because of a number of factors. One of which is the fact that fitness is really a property of an organism over its entire lifetime. It's measured both in terms of the number of offspring they have, but really it's a measurement of how many copies of their genome they manage to get into future generations. So you can get to fitness through multiple different methods, and realistically you would want to measure something over multiple generations, because perhaps by having all of these children, you're not actually very good at providing for any of them, and so you end up with fewer grandchildren because you focus too much on providing a large number of relatively poorly provisioned offspring. So, ideally, we would rather look at something where we can measure fitness across multiple generations in order to more accurately figure out what's going on. So I'm working with the E. coli experiment. So the long-term evolution experiment is a project that's been going on in Rich Lenski's lab since February of 1988, so we recently had the 25th birthday of the lines. There are 12 populations founded from an initial clonal ancestor. Half of them are capable of using a particular sugar called arachidose that doesn't actually exist within the media we give them, and it's completely neutral, but it's useful as a marker to tell the strains apart. So half of them are capable of growing on it, half of them are not capable of growing on that. We have daily transfers of 1% of the liquid volume of each one of these separate populations, growing separately from each other. That allows us to control the number of generations that are experienced within any one of these flasks, because they have the log base 2 of 100 generations every single day. Now this will remain true even if the carrying capacity of the cultures changes due to evolutionary change within the populations, because while there may be slightly more or slightly fewer generations within that one particular day, the fact that you have this 100-fold dilution means that they grow 100-fold every day based on whatever you dilute them down to. So we go through 6 2/3 generations a day, which means that we're over 50,000 generations. Actually, right now we're a little over 57,000 generations, depending on which one of the 12 populations you are. Some of them are a little further behind. We have frozen samples of these from every 500 generations, which is incredibly useful for analyzing long-term trajectories in the data, because I don't have to rely on measurements someone else took 20 years ago. I can go back to the freezer, revive populations from 20 years ago, and ones currently, and compete them head-to-head and figure things out that way. So because I'm really worried about the fitness in these populations, have to tell you a little bit about how we actually measure fitness. So all of our fitness measurements are done as competitive head-to-head fitness measurements, rather than a lot of people use various different proxies for fitness. In higher organisms, you might use seed set or number of eggs produced or some metric like this. A decent number of microbiologists actually just use maximum growth rate, which is a large component of fitness, but not quite everything. We are actually going to end up calculating the realized Malthusian parameters, which is essentially the number of generations that one population goes through within the course of our competition compared to the other population. So we grow up two different populations, we incubate them separately from each other for a little while just to get them used to the conditions of our competition. We inoculate them together and run a dilution series, and because we have these differently marked strains, we can tell which of the colonies came from which of the two different initial populations. We incubate that for 24 hours and do a dilution again. And we can count the number of colonies produced in each one and use that to figure out how many generations each one of them went through over the course of this competition. Most of our competitions are done as one-day fitness competitions. Sometimes we need a little more precision, so we can keep going further out. The more generations you include within the competition, the more precision you get. However, you lose the ability to measure things if fitness differentials become too large, and eventually, if you run your competition too long, you have to worry about evolutionary change within the course of your competition. So,
do you ever worry about like survivability in stationary phase?
That's not really a concern with us. We have checked in the past to see whether we have any appreciable depth within the 24-hour time frame we have. In short-term stationary phase and highly buffered media, such as we're using, we do not see any appreciable depth in E. coli. E. coli handles short-term stationary phase just fine. Other microbes and certain other media types, we would have more of a problem for it, but since we don't have really appreciable depth in the ancestor, we're not really expecting it to evolve over the course of our competitions. So, major questions that I'm addressing here are, does fitness actually continue to increase over extremely long periods of time within our populations? What is the shape of this trajectory in fitness? This can tell us some ability in what we can use to predict future values. And what can changes in fitness tell us about the adaptive landscape? So the adaptive landscape is a common heuristic tool in thinking about evolution. I'm not really going to get into a lot of detail about, there are a lot of arguments about adaptive landscapes and whether things are rugged or peaked, and it's difficult to think in more than 3 dimensions, so it's hard to tell some of these things, but we'll discuss a little bit of that later. So starting now, does fitness continue to increase? So I can take populations from different time points. This is a type of graph I'm going to show a couple of later on, so I'm going to talk you through what this actually is. Along the x-axis, I have fitness at one particular time point. In this case, it's at 40,000 generations. Along the y-axis, I have it at a different time point. It's always going to be an earlier time point along the x-axis and a later time point along the y-axis. And I have here the y equals xy. So any of the points that manage to fall above and to the left of the line are going to be more fit at the later time point. Anything that is below and to the right is going to be more fit at the earlier time point. Now I have 12 populations of E. coli. Unfortunately, I can only work with 9 of them reasonably at this particular time point. 2 of them stopped growing on our standard competition plates, and so we have other metrics of measuring their fitness, but it's easier to just show everything from the same sort of measurement, so we don't have bias introduced on the type of measurement being used. And one of the 12 populations we've heard about in great detail from a number of other people in the lab, particularly Zach and Caroline. It's error -3. It's the one that's capable of using citrate. It has a much higher fitness compared to the other ones at these time points because this is after the citrate expansion. So we can't do competitions for quite as many days. We can't get the same level of resolution. So of the 9 populations I do have, though, I competed all of them against clones at 40,000 generations in order to get more precise measurements. These are actually multi-day competitions. 95% confidence intervals are shown with the error bars. You can see that these points in general are falling above and to the left of the line. If I do a one-tailed t-test on that, I find that this is in fact significant. Populations are improving in fitness between 40,000 and 50,000 generations. On average, it's about a 3% relative gain over those 10,000 generations. So, we have evolution going on. Now we can start looking at all of our individual populations. So here, all of these points are the means for each individual population at a given time point. So everything was done in replication. There are 41 separate time points across here. This is giving us the general cloud of all the data. However, it's more useful to think in terms of the grand mean. So this is taking the means of each of the populations, figuring out what is happening overall to the populations as a whole over time.
So you're not modeling each population separately?
At the moment, no. At the moment, I'm looking at just the grand means. I have information about individual populations later. By taking the mean of each population and taking the grand mean of that, you don't have to worry about whether individual populations are having greater or lesser leverage due to particular missing values. Beyond the fact that 3 of my populations I have to truncate because they stopped growing reliably on X-standard competition media at certain time points, there are a total of 5 missing values otherwise in the data set. So here again is just all the grand means. And in the past, our lab has tended to use this particular mathematical model to fit what is happening in fitness over long periods of time. This is a rectangular hyperbola. It has a couple interesting properties in that it tends to have rapid increases relatively early on, followed by a gradual slowdown. One of the potential problems of this model, however, is that this has a hard asymptote. As time goes out towards infinity, the relative values of these things will drop out, so eventually it's just the A value that contributes to what is the maximum fitness that you can ever get from this particular model. Well, back when I started doing this, one of our lab's major funding sources was actually from DARPA, who were trying to have us figure out what the fundamental laws of biology were. And the idea was for me to fit a power law to evolution, because power laws explain a ridiculously large number of things that we have no particular reason to expect a power law to fit. So the idea was that I was going to fit this particular function. So this is a simple power law, same number of parameters as we were dealing with before. It does, however, have this slight offset because otherwise you can't take the log of 0 and you end up having to throw out the ancestral data that we go in great detail. Now the problem is between these two models, it turns out that the hyperbolic model is actually a substantially better fit. The difference in BIC value here of greater than 90. In general, differences in BIC value between 2 and 6 are considered moderate support. Between 6 and 10 are considered strong support. Anything greater than 10 is considered very strong support. So it would look like things would be done here, except for the fact that I actually made a mistake. So this is the model I was supposed to be fitting, but I misread the email and put the parameter in the wrong location. So I instead ended up fitting that model by pure chance. This model, however, is substantially better than the hyperbolic model that I was supposed to be fitting. Difference in BIC value of 33.7. That corresponds to a difference in the posterior odds ratio of about 20.8 million. So, now it turns out that these models are actually very close to each other. The only reason why my log time model, which is what we're now referring to this as, is different from the power law is because of this internal offset of 1. If you get rid of that particular value, if you get rid of the adding 1 to both the log time model and the power law, they end up becoming formally mathematically equivalent because the b value outside is simply a times log of b, nice properties of logarithms. I can get into that in detail if anyone actually cares. But, by chance, I now have a much nicer model. Now, this tells me information about what is happening at the grand population mean. The question is, this might simply be an artifact of the fact that we're taking these means of populations. What if we look at individual populations? So, I have this table here, summarizing all of that, ranked in order from the differences in BIC value here. So things in blue are better fit for the log time model. Things in red are better fit for the hyperbolic model. Now, These ones in gray are things of small differences in BIC. These are not considered to be meaningful differences. These ones here are considered to be moderate support. These are strong support, and these are very strong support. So overall, this pattern is not simply being driven by a very small number of populations. In fact, more than half of our populations are showing support for the log time model as opposed to the hyperbolic model. And when you then take into account the degree to which there is support, it overwhelmingly tips in favor of the log time models. So, there are other things that are beneficial about this log time model beyond simply the fact that it fits our current data better. Because my interests here are primarily in predicting evolutionary potential and what the responses are going to be. But because I have this large data set, I can use earlier subsets of the data to predict what is going to happen over longer time frames. So here I've taken the hyperbolic model and fit it for just the first 5,000 generations of data I have. And as I add in more data, the line changes to some extent. So here I go up to 10,000, 20, 30, 40, 50. Every time I add more data in, I tend to increase what that curve happens to be. And you'll see that overall we still have this tendency that these later time points tend to be higher than what we're predicting from our curve. In fact, many of them are higher than what the asymptote calculated value would be. So by the time we find an asymptote, we find that we already have generation data that is beyond what that asymptote is supposed to give us. On the other hand, if I look at the log time model, as I start adding in time points, as I get out here to 20,000, things have moved around to some extent. Then I add in 30, 40, 50. I have exactly the same curve as I add in more data. So using just the first 20% of the first 40% of generations, I'm predicting the same thing I would get from all of them. Where between 10 and 20,000 generations it stops moving around, I haven't actually gone through and analyzed yet. That's on my list of things to do. Now this is true for the populations as a whole. What if we start looking at individual populations? So if I'm going to pick just one individual population to show you at the moment, I'd rather do so in a somewhat systematic way. So I'm going to use here era plus 3, because it's one of the median values as far as the benefit of using the log time model versus the hyperbolic model. It also has the advantage that it didn't have to be truncated, so I have the full 50,000 generations on our standard computation. If I go through the same set of slides for this particular population, we have each individual replicate of working with the population. We then get the means of all of those individual replicates. We fit the two models, and as you've seen before, it's a BIC value between 2 and 6, so it's considered to be moderate support, but not particularly strong. And as we add in more data, we continue to move around with our hyperbolic model. Things are constantly changing, but we also systematically have issues, particularly at the later time points with the hyperbolic model. On the other hand, with the log time model, as we add in more generations, we don't really change this curve. So this is a general property that we found across these populations, and it's true not only for the grand mean, but it is also true within many of the individual populations. So we've shown for this particular set, log time fits the current data considerably better. It predicts the future data better. It has this aspect that it doesn't actually have a hard limit, so we can predict things will keep changing over vast periods of time. This is true for both the grand mean and the individual populations. So next we wanted to start looking at whether this is being caused by different artifacts in the way that we've been doing our sampling or due to artifacts within our particular populations. So, for instance, here with our different populations, we can say that, well, we see that 3 of them had to be truncated for specific reasons. Here, era plus 6, it actually stops growing reliably on our standard computation media after 4,000 generations. I have data from it on other ones through an alternate method. That data is not actually included in here. Similarly, era minus 2 stops at around 32,000 generations, and era minus 3 has the citrate explosion, so I'm unable to get it after about 32,000 generations. So what if we remove those ones? Well, from looking at this table, it's not surprising that that probably wouldn't make a big difference because these are things that don't have particularly strong support in either direction. But if we take a look at things, here's my grand mean from the 12 populations. Here's my grand mean for just the 9 populations that didn't have to be truncated. So it lowers the BIC value a little bit, but it's still a vastly better support for the log time model than for the hyperbolic. Well, another thing that was brought up by a former member of my committee is the fact that our sampling regime here is not completely even. At these earlier time points, we have more dense sampling. For the first 5,000 generations, I have every 500 generations as a sample. Then between 5,000 and 20,000, I have every 1,000 generations as a sample. And beyond 20,000, I have every 2,000 generations as a sample. It's easy to focus on differences between things that have asymptotes and things that don't, but that's not the only possible difference here. It could be that both of these models are doing about the same job towards the end, but just that the log time model is doing a much better job earlier on, and since we have denser sampling earlier on, that could be driving the differences in BIC values. So, if I instead go through and remove about a third of my data, so I've got only every 2,000 generations evenly distributed all the way across, I still have significantly better support for the log time model than I do for the hyperbolic. And that holds true even if I drop out the 3 truncated populations. So next we wanted to consider whether the possibility is that this is all being driven by the fact that 6 of our lines become hypermutators. These are lines that have drastically increased their mutation rate relative to the ancestor, typically through mutations that occur through the methyl-directed mismatch repair system, which is a way that cells have of figuring out when there's a mismatch between the bases on 2 different strands of DNA, which is the original one and which is the newly synthesized one that's the one that's probably in error, and you correct that. So, if I drop out all of my populations that eventually become hypermutators, I now have only 6 populations that I'm looking for. I still have substantially better support for the log time model. If I then take through and take only evenly distributed values here, I'm now left with approximately a third of the data that I started with. And I still end up with strong support. No longer quite as strong, but these are still substantial values in terms of differences between the BIC values. So it looks like that this is not being caused by artifacts related to the specific generations that I happen to be sampling, or the fact that some of our populations we don't have quite as completed data sets, or some of them change their mutation rate, and things that have a higher mutation rate are therefore potentially going to have radically different sorts of fitness changes. Great. So I have this sort of information. What else can I do with this? Well, I can start looking at correlations in fitness through time. Now, this is a more complicated graph than you need to actually pay attention to at the moment. Only pay attention to the values above this diagonal. So this is a heat map of correlations in fitness. So each individual column and each individual row is one particular generational time point, going from generation 500 up in the upper left down to generation 50,000 in the lower right. In this value above diagonal, anything that happens to be a negative correlation has been coded here as white. There are no more of those than you expect by pure chance. Anything that has positive values is color-coded in a heat map going from light yellow are low values of correlation to dark red are the highest correlations. Things to pick out from this are that the vast majority of these correlations are constant. These correlations persist over very long time periods. And that the darker colors generally happen closer to the diagonal, as you would expect. So these are things that are closer generation samples. You would expect more correlation in fitness. But we can get at that a little more simply by fitting our data based upon the differences in generations between 2 generations that we're looking for a correlation in. So over here on the left, we have things that are separated by fewer than 2,500 generations, all the way out to things that are separated by more than 32,000 generations in fitness. And so you get this nice gradual decline. So we have a decay of correlation over time. But these are still relatively meaningful values in that we have correlations on the order of 0.25. Correlation values are necessarily constrained between negative and positive 1. Yes?
So I'm just curious, in a given generation, do 5 replicates in the same lineage. So technical replicates, what are the correlations?
I have not run those numbers.
And so where they fall in these ideas?
I haven't run those numbers, so I don't actually know. These are correlations simply between the means of the biological replicates, but I need to check the technical replicates. This is analysis I've done in the past 2 days. Right. So we have this decay in correlation over time, but it's still relatively meaningful values, potentially, depending, of course, on where our correlations are from technical replicates. We also can look at variance over time. So one thing we had seen before is that the among population variance components, which is the AP.PC, we knew from earlier data in the long-term lines were increasing for some length of time. We didn't really know how things would go over long periods of time because prior to me doing this work, the last time anyone looked at any detail at fitness in the long-term lines was at 20,000 generations, and the last time anyone published on it was at 10,000 generations, so some time in the past. Now, the way the among population variance components are calculated, it's actually possible to get negative values because you're essentially partitioning variance within populations and among populations, and you have to get to a total value, so you can end up getting negative values for these things. The point, however, is merely to check whether this variance is in fact continuing to go up, because this is something that can tell us indirectly plausibility of certain arguments on adaptive landscapes. The idea here being that if you have populations that were exploring a single peaked landscape, you'll have variance between populations in fitness go up for a while, but eventually as they start to reach the top of that peak, that variance will start to drop back down as the values start to converge upon each other. On the other hand, if you're exploring a multi-peaked landscape, you'll have variance increase for a while as they go off to different peaks, and then as they reach the top of their peaks, they'll stay at the same variance. And if they haven't reached their peaks, they'll probably continue going up. Now, all of this is a plausibility argument. None of this is actually definitive. But we do, in fact, have a positive value for our variance over time. We can only find meaningful results at the moment of a linear fit. I've tried various higher order fits in all of them that I've tried so far. The linear comes out as significant, the higher order values are not significant. So we're not actually doing a terribly good job explaining it. Our squared values are pretty bad. But this is coming out as a significant increase, which simply means that we are increasing in fitness. We're increasing in variance. We're just not very good at figuring out exactly how that variance is increasing. We don't have a good mathematical model for that. This, however, is consistent with this being a rugged landscape in some sense of the word. Now, it's possible that this is in fact a single peak landscape because n-dimensional space, it's difficult to figure out how many peaks there could possibly be. But in terms of practical applications, what matters is that this variance is still increasing, and so therefore things are not really anywhere near top of the peak. Part of the reason we care about this, though, is that many ecological models assume that anything found out in nature is sitting at the top of the fitness peak, unless humans have very recently come in and disturbed things. Here we have populations that have been evolving for a very extended period of time in isolation from anything else, dealing with a standardized set of nutrients and environmental conditions, and not having to worry about predators or prey or most types of co-evolutionary dynamics outside of interactions with their own very close kin within their classes. If these populations are still increasing significantly in fitness, and different populations are increasing their variance relative to each other, it makes it less plausible that we can just assume that everything out in nature is already optimized, because these are things that are very slow optimization. So we can find from this information that correlations do persist over long periods of time. Correlations decay over time. So historical contingency is important, but there's more than just historical contingency at play here. Because if it was only historical contingency, we wouldn't really expect much of a decay. Once you have one population getting ahead, it will stay that much further ahead for the rest of the time. We do have populations moving in relative rank order to each other across the course of this experiment. We have variance still increasing, which is consistent with multiple peaks, but we cannot actually rule out the possibility that they are climbing a single peak. They're just so far from reaching the top of it that it doesn't matter that there's only one peak. Right. So, this is information information I have from everything of individual populations competed against the ancestor. But that's not the only way we can do competitions. So here, for instance, I have in cyan one of the populations with error -1, and in brown with error +1 a different population. These are the log time model fitnesses, and the dashed lines are the bootstrapped 95% confidence intervals, so you have some idea what's going on. From the individual populations against the ancestor, we can figure out what we expect the ratio of those fitness values to be relative to each other. So the green here is my expected error -1 against error +1 if they're both drawn from the same population. The same generation. So I can put in here a bunch of fitness measurements I did of doing those competitions. For error +1 and error -1, I have a much denser sampling than I have for anything else as far as these pairs go. Here are the means of all of those particular values, and here we can scale in to see that in a little more detail. So for error +1 and error -1, I have actually competitions done between the pair of them in replicate at every 500 generations for the entire set. And I have these done as 3-day competitions so I can get a little more precision in what our fitness measure is. Here I can put up all of the mean values, so that's what we're trying to fit. And here is the relative fitness of one line. So we can see here that error -1 does get ahead of error +1, but it seems to only get somewhere around 5, maybe 7% ahead, and then stays relatively consistently that far ahead. Now this is what we would have projected based on just the information from the individual populations against the ancestor. So you can see that many of the points do fall within that, but clearly the shape of that is not fitting our overall shape terribly well. So instead, if we use some sort of locally weighted regression factor, so in this case I'm using the lowest function in R, I can get what we expect the ratio is between these two based on weighting it by local points, essentially. The confidence intervals on this, I am in no way confident that I have done that correctly. These are bootstrap confidence intervals, and I don't yet know whether bootstrapping is an appropriate thing to do with the local estimated weighted regressions. I need to dig into more detail on how that's actually done to know whether that is an appropriate way to do it. I just wanted some form of error bar to put up for the moment. Giving you all my caveats up front. But you can see that this actually is doing a much better job of describing what the overall line is doing. We get this difference between them that does seem to persist over time. I have these values again for all of the natural pairs of populations. I think there's a limit to how many graphs any of you want to see during one presentation, so I can show them to you in other slides here later. Right. So from the long-term fitness data, I have that this log time model is our best model that we've found so far for both fitting our current data and for predicting our future data. If there are peaks, we have yet to reach them. There are a number of possible reasons for this. One could be that we simply haven't had enough time. Despite the fact that this has been going on for 25 years, we have gigantic population sizes. And gigantic population sizes mean two things. One is that we have a lot of clonal interference. So those of you who aren't familiar with clonal interference, this is largely something that occurs in asexual organisms, where if you have two competing clades that both have beneficial mutations in them compared to the predominant clade within the population at any time, without recombination, you can't easily get those two mutations that are both beneficial onto the same background. So one of them is going to win out over long periods of time. But it's competing not only against the general population that's better than, but also this subpopulation that it is somewhat better than, but which is itself better than the overall population. Because of the fact that we're dealing with an asexual organism, most of our sweeps are going to have to be sequential rather than things combining together. That means that, for instance, things that are highly visible to selection, things that are more than 4 orders of magnitude above the limit of visibility to selection due to our gigantic population sizes, are taking 25,000 generations to sweep. So we haven't even come close to exhausting the beneficial mutations available. We also have evidence from a number of lines of frequency dependence. This can be seen from potential cross-feeding between subpopulations. It can also come up for a number of different ecological reasons. But whenever you have frequency dependence, you have the possibility of slowing down the total adaptation in the system, because as an individual clade gets driven to lower and lower values, it becomes more and more fit, and therefore you can't get rid of it, and things will persist over very long periods of time. We also have epistasis, which we always like to invoke on one of the reasons why evolution is not a completely trivial process. Things that are beneficial in one background are not necessarily beneficial in other backgrounds, and things that are individually beneficial are not necessarily beneficial in combination with each other. All of these things can end up slowing down how rapidly you get to the top of the peak in the fitness landscape. However, because of what we currently have, long-term predictions are possible. As I've shown, using less than 40% of our data, I can almost exactly predict what we're going to get using the full set of the data. So this allows us to make predictions for longer time periods in the future than we ever could have with previous models we used. But that's great. This tells us about one particular system. What else can it tell us about? And because I'm in Beacon, it is not at all surprising that my next system that I look at is Avida. I will tell you up front, all of the Avida work I'm going to be talking about right now is in collaboration with David Bryson, who's a former student of Charles Sofria's. Dave ran a bunch of very long runs in a couple of environments for things related to his thesis, but because he ran these giant long runs and has data from them, I can then analyze what's going on in fitness across these populations over ridiculous timescales. For those of you who aren't as familiar with Avida, basically it's a digital evolution platform where you have a world. In all of the cases I'm talking about here, it's a toroidal world, potentially 3,600 organisms. Each one of those cells can contain a circular genome of a self-replicating computer program. These computer programs are competing over resources in the environment, generally CPU cycles, which they may get by doing various different tasks. Although in one of the environments I'm going to talk about, there are no rewards. All they can do is get slightly better at being efficient replicators. There are all sorts of new environments available in Avida. Most of you who have heard about Avida have probably heard about the Logic 9 environment, which is based upon 9 particular logical tasks that can be done on pairs of binary numbers. I'm actually not talking about the Logic 9 environment. I'm going to extremes from either direction of that. So first I will be talking to you about the Logic 77 environment and then a no-reward environment. So the Logic 77 environment is a much more complicated one, that it gives rewards potentially for any 1, 2, or 3 input logic function. But unlike in the Logic 9 environment, more complicated tasks do not actually result in any greater reward than simpler tasks do. Every task that you complete effectively raises your fitness by a factor of 2, so we're looking here at log 2 of fitness because that gives us the proxy for number of tasks, and also so that way individual runs that are way off the curve from everyone else don't end up making it so you can't see anything that's going on. Now, how long we run these things is a variable of time. This is the length of time of a fairly standard Avida run. It goes out to 100,000 updates. It looks like things have basically leveled off. You're not getting much improvement. But this is a subset of the data he gave. So what if we go out to 250,000 generations? And as I'm going through these, every time I go out to a new generation, everything that was previously on the graph is going to be over here to the left of whatever the dashed line is on. So that way you can see how things are scaling. Now if we go out over longer periods of time, we have fitness continuing to improve, but it always looks like it's leveling off. But it never actually is leveling off. So here we're at 1 million, here we're at 2.5 million, here we're at 5 million, now we're out to 10 million updates. Every time it looks like we have a plateau, that plateau is really only a temporary feature. And if you go out and zoom out to longer time scales, it turns out that it's not actually that. Right. So, the first thing to do, looking at all of this data, is the same sort of, yes?
But you have a max fitness of 77 here, right? Of log 2 of 77.
Essentially, although not entirely.
Okay.
So 77 is essentially as good as it can get because that would be if it did all the tasks, but it can then still optimize its replication rate within a set of tasks. And so it's not quite bounded to 77, but 77 is close to saturation of this. So if I do the same sort of analysis that I started out with for the long-term lines, basically looking to see whether over long periods of time things are still managing to improve, I arbitrarily decided, let's look at 2/3 of the way through here. So, switch the slide. One thing I wanted to tell you at the moment, all of this stuff I'm talking about right now, I'm doing in terms of updates because the runs were set up to dump out information at specific updates and go to a specific number of updates. Here are the 20 different runs he gave me. As you can see, they reach wildly different numbers of generations, and when they output at a specific update number, each one of these populations is likely to be at a different generation, so getting grand means isn't quite applicable because things don't line up that way. So instead I'm doing everything in terms of updates just because that's the data I have at the moment. Right. So, if I look here at log 2 of fitness at 2/3 of the way through versus log fitness all the way through, again, anything that is above this y equals x line is going to be more fit at the later time point. As you can see looking at this, things in fact are more fit at later time points. I don't have error bars on here because one of the features of fitness in Avida is that it is actually explicit rather than measured in terms of us doing experimental noise measurements. So we have that. Look, it's significant. Not terribly surprising. This is a one-tailed t-test. It's all very simple. Right. I can also run a linear regression on whether there's an overall linear increase in fitness over that time period. You can see here significance of the linear term, 3.6 times 10 to the negative 14. So yes, fitness is in fact increasing over very long time periods in this particular environment.
But you're not accounting for very much variation.
It's less than 2%.
Yes, this is actually doing a very bad job of figuring out what's going on. I'm not sure at the moment how much of that is due to the fact that we have radically different fitness regimes, and so I think in general this is not actually a very good model, but it was merely showing whether there is an increase or not. If I look within each one of the individual 20 runs that this was done for, 18 out of the 20 give me significant improvements over this later time period. So there is still improvement going on. The fact that this is a terrible R-squared value may be due to the fact that there isn't much of a change going on, but it also may be due to the fact that the change is not actually linear.
Right, and so I can't remember, are you estimating originally were we looking at log fitness? Oh,
Yeah, I'm looking at log fitness because otherwise if I'm working with absolute fitness,
Right.
really won't. Right. So, one thing to note here, however, individual curves here, the vast majority of individual curves are actually better fit for the hyperbolic model than they are for the log time model in this particular data set. It turns out that the better log time fit is a property of the set of populations as a whole, but it is not generally true in individual populations, even though individual populations are still increasing. I have reasons for this that I will be discussing shortly, but wanted to talk to you about the fact that here's our grand mean. This is, again, the hyperbolic model. If I try and fit everything, you'll notice that as with all the other hyperbolic models, it has this region where it overestimates fitness, then over the long periods of time, it ends up underestimating it. This is the log time model fit on that. And because when you put the two of them together, and since we have such an incredibly dense number of updates giving us values here, difference in BIC value of 8,551.76.
Barely.
That is a difference in posterior odds ratio of e to the 4,276. So rather big numbers. Right. So going to the other extreme, what if we give them no tasks? So part of this may be driven by the fact that clearly there's a lot of room for improvement from a fitness of one, essentially, well, beneath one of the ancestor, to a fitness of somewhere out in the log to 50 to 60. So if we give them absolutely no tasks, we start with an ancestral organism that is capable of self-replication. We then let it go in an environment where the only thing it can do to improve its fitness is become slightly better at replicating. We let it go for 6 2/3 million updates. We then check it against what it is at 10 million updates. There is, in fact, significant improvement over this time period. Yes, I have stats that are significant. If I were to do the linear regression on this, same as before, these are not log transform data. I was choosing not to log transform because we're dealing with such a narrow window. Part of the reason I was doing the log transformations earlier was it was necessary to get my residuals reasonably distributed.
How small did it become?
So the one of these that I looked at in detail, I believe it had a total genome size of between 30 and 40 because there's a minimum requirement or else they're considered inviable. But it actually, the entire organism was a copy loop in a bizarre logic flow. It would attempt to divide, but it would only manage to divide if it had exactly copied a certain number of instructions. There were 11 one-step mutations that were beneficial, and of those 11, all of them were insertions and deletions. There were no single-step substitution beneficial mutations.
So it's unrolling the loop.
Yes, basically. It turned itself into the loop entirely.
So it looks like at 10 million updates, though, there's no variance left. Virtually no variance.
Yes, there is very little variance left in fitness between the different
So
populations.
do you have it at 11 million or whatever?
No, these were runs that he ran for 10 million updates. These runs going to 10 million updates take about a month.
So
on each
if
CC.
you did 3,333, yes, that, you know.
Yes, 1/3 of the way through.
Yes. You have this, essentially the same amount of variance as you see this 6666.
I have no idea. I haven't listed that. I picked these numbers just arbitrarily.
Okay, because it's amazing that you had so much variance 2/3 of the way through, and then I mean, just I guess it's blacked out.
Yeah. Also, because in the no-task environment, things are much more constrained to have relatively the same number of generations as each other, because you don't have these massive improvements in fitness that are affecting the number of generations. I also did these sorts of analyses for just basically taking out the first 500,000 generations. There are about 780,000 generations. So again, taking out roughly the first 2/3, and we again get very significant improvements, but really bad R-squared numbers. So we do get improvements over vast periods of time with Avida. However, this improvement being in terms of log time seems to be true of the data I've looked at the mean across populations, but it is rarely the case within individual populations. I think part of this is being driven by the fact that our ancestor is extremely unfit in these things, especially compared to what it is in the E. coli data. When we're looking at these things, the E. coli have been improving somewhere around 70% improvement in fitness. The Avida organisms are improving by a factor of 10 to the 18, so vastly different parameter scales. We will be looking at doing these in a slightly different way, but I think that's part of what's going on here. We have information that fitness does seem to be rising over extremely long periods of time. The log time, whatever logarithm we happen to be choosing, has no particular upper limit to be reached. So these would tend to predict increases in fitness over vast scales. Now, it's true that eventually in any physical organism, there has to be a hard upper limit. As I have argued in my committee meetings before, eventually you are going to run into diffusion constraints that even if you are perfectly using all of the resources available to you, they have to get to your cells to some extent. But as things slow down more and more as time goes out, because of the way logarithms work, it may be the fact that we'll reach the entropic heat depth of the universe before we actually reach the kinetically optimized constraint within our individual organisms.
Did you simulate the long-term lines or project into, where they would be when we if the heat depth?
So that depends on changes in parameter regime on when the transfers are being done. I have done calculations for if we treat the first 50,000 generations of long-term lines as one human generation. And we go through 50,000 human generations, what is our expected doubling time within our media out there? And then depending on what assumptions I make
What
about
are we looking at there, roughly?
well, so it would be 50,000 times 20. So we're out a million years in the future. Depending on what assumptions I make about the inherent lag time within our populations, our doubling time gets down to somewhere in the neighborhood of 12 minutes in our media, which is fast but not utterly ridiculous. Yes. So these are properties that seem to be true in very different sorts of parameter spaces. So I have other things ongoing. We're actually in the process of doing some runs in Logic 9, and we're doing output by generation rather than output by update because I'm a biologist and I prefer thinking in terms of generations than updates. So it'd be nice to look at things that way. I think part of the problem with the Avida stuff is that our ancestor is so incredibly poorly adapted. We're actually going to be using a somewhat evolved ancestor to get things out to a higher starting ground.
And you could decrease the fitness gain for getting any of the single.
That is another way.
Don't double, give it 5% advantage.
Yes.
Similarly, would you take out is the gauge each step additive, not this exponential, sometimes.
Yeah, the only environment I have data from at the moment, everything is essentially multiplicative. Every time you get a new task, you double your fitness, more or less. You actually double your merit, technically, rather than doubling your fitness, but close enough for what we're talking about. If anyone has any other long-term resets on good things. Otherwise, I have acknowledgements. So we have up here on the upper picture is the Linsky lab, obviously run by Rich. The lower picture is the Afria lab, run by Charles. Particular people to point out are Nuja, who is our lab manager and keeps me supplied with all of my flasks and iPad tips, so I don't spend my entire life dealing with that. And Dave Bryson over here, all the way on the left on the red shirt, he's the one who actually ran things for the Avida data I have. Members of my committee who have been talking about this one, obviously, in addition to Charles and Rich. And other than that, I would be happy to take questions. Yeah.
So, two thoughts. One thing for the long-term lines, you can, because you have the 9 line, you can fit sort of essentially a random effects model for that. So you'll basically look at the variance among lineages directly. So instead of having to build either 9 models, so the no pooled on one extreme or complete pooled on the other extreme, which is what you did, you can actually model that explicitly. So
yes.
The other thing is, because the linear models that you fit probably are, because fitness at one point is correlated to fitness at another point.
Yes.
They're not, there's assumptions you're violating there that are important, but they're easy to model.
Okay.
So
I know that there are assumptions I'm violating there. I just wanted something quick and dirty to see whether things are increasing. I know that there are problems with that, but I'm not sure how to properly do
straightforward.
correlate stuff. Yes. Any other questions? Yes. Ben.
Question from Washington.
Yes.
Just curious about some of the differences in the individual populations between Avida and the coli lines. It seems like you're getting sort of better hyperbolic fits. I'm curious so you talk about landscapes, and generally we think of these things as static structures. And I'm curious if in the way that the Avida runs were executed, that sort of is a better approximation in some sense, whereas as E. coli evolved, it presumably could change its environment. You talk about crossfeeding and other things, making sort of types that in a non-changing environment might not be sort of available. What sort of role does the organism, and these things could be built into Avida, so if you were looking at parasites or resources generated by the Avidians that other Avidians could use, you could get maybe some of these niche constructive things working there as well. Do you think that this has any role in sort of getting a better log fit in the long run over hyperbolic?
So first of all, short answer, yes. In more detail, we were actually discussing this earlier this week. Part of the reason why we don't get the sorts of interactions among organisms within Avida in these particular runs is these are things set up without messaging turned on. None of them are outputting partial numbers that are available for other ones to potentially use. We don't have parasites in the system. So a lot of the things that people have implemented in order to look at interactions between organisms aren't on in these particular runs. So we don't expect the same sort of frequency dependence within these runs as we get within E. coli. I do find it interesting that the overall grand mean population for the Avida stuff is still giving us these sorts of log time fits the same way we're getting it within E. coli, even though within the individual populations a lot of it is not happening the same way. I think the frequency dependence is one of the potential explanations of that. I think a larger explanation at first pass is going to be how incredibly unfit the ancestor is. So that's one of the things we're hoping to look at.
Well, population size may be something
Yes,
else.
population size is also a huge factor because these things are limited to our Avida population size is a maximum of 3,600, whereas our E. coli population size, the smaller size of the bottleneck, is still something times 10 to the 7, in our particular cultures. So you have drastically different population sizes, which influence a combination of traits, including adaptation rate, probability of loss, the fact that the E. coli are actually going through these serial bottlenecks, and so you get a lot of things that are getting lost because they're simply being diluted away. That becomes potentially a larger factor in certain portions of the parameter space than you get with replacement within these particular Avida runs. You end up with differences of even what is visible to selection, and of course the fact that every individual new task that's being gained within this Logic 77 environment essentially doubles fitness, whereas there are no single mutations that double fitness that we have in the long-term line, with the possible exception of the citrate stuff, but that's actually a multiple mutation complicated story.
I mean, you see that in many of the Avida runs, they were stuck in local fitness peaks and had to wait for a double mutation or more in order to cross the peak. So the beneficial mutations were difficult for them to get simply because the population size was so low. But if you're putting the 20 runs together, basically you're going to get these beneficial mutations in a sense somewhere in the population, in one of the 20. So it's like clearly the grand mean acts as if a much larger population, and therefore in fact you're going to see these.
But only to some extent.
Only to some extent.
Because you don't have movement across the population.
That's
so
right.
Something that sweeps one population will not necessarily go into
Yeah,
another.
you would probably have to have a much larger population for a single Avida run to have this type of larger than 20 times. But if you would have it, let's say 1,000 times, it's not impossible that different areas in the sense of the population would become individual subpopulations, and you would see the beneficial mutations.
That is certainly plausible. Our ability to actually test that is limited to some extent by computational resources.
Right. However, Dave Knuster actually
Yes,
said you
I know Dave is
could
working on it.
potentially run a billion or so. It's not quite, 10 to the 7, but
Yes. It's still
Actually,
much larger than it is.
that's
Right around 10 to 7.
yeah. So 10 to 7, I think you can even do today. To 10 to 9 is what they do.
Yeah, I was wondering about the citrate guy. So is it tracking along and then does it just jump or
So
what happens?
that depends on in which way you're looking. So if you look at it in the individual fitness against the ancestor, it looks like it's going up for a while. There's not really a sudden change as it crosses the ability to use citrate, and then it starts to come back down later on. On the other hand, if we look at it as an individual pair, something happened somewhere between 32,000 and 34,000 populations. So this is era minus 3 competed against era plus 3. I'm not entirely confident of some of these numbers up here because my number of colonies on the plates dropped a lot lower than I'm used to using. And so these are the 3-day fitnesses because I just wasn't thinking about the fact that I needed to plate things at earlier time points. So I'm actually in the process of redoing these ones. I'll have these done next month. But in competition against another evolved line, there is a sudden spike in fitness, but when you look at it against the ancestor, that is not the case.
Any other questions from non-MSU places?
So a question here from Idaho on that figure that you've got on there. So you competed the error minus 3 lineage against the error plus 3 lineage, right?
Yes.
And that is the outcome of that experiment. If you were to repeat that, say, error minus 2 versus error plus 6, do you expect that same outcome? Or is this outcome unique or...
That's magic. Was that a plant?
I don't have minus plus 6 because those are the 2 strains that stopped growing reliably on TA. So I made an unnatural pair out of the other one that's left over from each of the 2. But for all of our other pairs, we get differences in fitness that are generally relatively close to 1. Sometimes things go as much as 5 to 10% different from each other, but it's only really the error -3 that drastically takes off compared to someone else.
Yeah. Okay.